Okay, this is a redo of the quick little lecture I gave on Wednesday. So this is your intro to the paper. I've added some figures from the paper to help clarify what I'm talking about. So we talked about vaccines, and we talked about how influenza is constantly mutating and changing either through antigenic drift or big changes through antigenic shift. And so a big goal is to find a universal flu vaccine, something that's going to protect you against multiple types of influenza, both the group 1 and group 2, type A and B, last long enough for a flu season, at least, although I would think that if it's really a universal flu vaccine, should last a lot longer, be suitable for all age groups, and really also immunocompromised people. Oops. Okay, we'll go there. So what I have been reading over the past couple years about a universal flu vaccine is the idea is that we're trying to get the body to generate antibodies to conserved regions of influenza. Because if you keep making antibodies to the ever-changing regions, or you make a vaccine that stimulates antibody production to the different regions, you're not going to have a universal protection, and you're going to be recreating a new flu vaccine every year. So the papers that we've gone through in other virology classes over the past couple years has all been looking at antibodies to the stalk, which is the more conserved, so stalk or stem region, because even as the head here changes, you still have antibodies to the conserved region. That's kind of the big picture goal. This paper is actually not a vaccine paper, but talks about universal protection by creating these antibodies again to this more conserved stem or stalk domain. So let's review what antibodies are. So this is an example of a human antibody. So you'll see me draw it like this kind of goofy Y to represent the heavy and light chains and how they're put together. This top region, and especially here where you see the V for variable, this is the region that is unique to each class. I don't want to say class. Each kind of, I don't want to say group either, but I'll explain it better in a minute. So each kind of group of antibodies. So this variable region is different, and it is the antigen binding region. FAB stands for fragment antigen binding. So this is what gives specificity. So you have to have antibodies that are specific to a specific antigen, something they recognize. And so we have thousands and thousands of different antibodies floating around in our body that all have different variable regions that are very specific to a specific antigen. Okay. So like I was saying before, if it's specific to the head group, then the antibody for an H5 head group is not going to react with the H8. And likewise, But here, if we can get antibodies to a conserved region, they could bind and block infection of multiple types of influenza. Antibodies are produced by B cells that are actually called plasma cells. And this is part of the memory of your adaptive immune system. Okay. So when you get a vaccine, you're hoping to stimulate antibody production through these plasma cells and keep these around for a long time so that you are ready for a challenge with the virus or an infection, ready to block it. The region down here, this FC region, this is the part that interacts with other cells and cell receptors. Okay. Some of the terms you'll see in the paper. So I just put this up here just to let you know we have multiple different types of antibodies. The IG stands for immunoglobulin. Okay, so all of these are called IG, and then you have the different subtypes. Okay, so they'll be talking about the IgG, which is composes about 80% of all antibodies. We're gonna represent antibodies as AB. It's the main antibody in your blood, so it's circulating around, kind of as a surveyor, to find antigens that it is specific for. So this paper introduces this idea of single domain antibodies. Okay. So we have multiple domains here, or two, and we have this heavy and light chain structure. So these antibodies from the Camelidae family of things like camels and llamas, they only have this single domain for recognizing antigens. It's a much smaller antibody, and so what we're going to see is that the researchers take advantage of this and create bigger antibodies by fusing some of these single domains together. So each of these numbers represents a single domain antibody that they selected for as good at binding to and binding to the stalk region of influenza. What they did in this paper after they identified 4 of these single domains is they molecularly kind of linked them together. To create what we call MD, multiple domain antibodies. Okay. Then they took it a step further and put the FC region of IgG onto basically like 2 chains of the multi-domain, and this looks more like a mammalian antibody. So not only does it have the antigen binding, but it also has the FC region for cellular interaction. And the CR antibodies that they're showing you here, these were antibodies that have been isolated and naturally produced from the influenza vaccine. So these are like your control. These are antibodies we know works so they work to block influenza infection. And you'll see that a lot of the paper compares the neutralization activity of these multi-domain antibodies versus a naturally made antibody. A couple things to remember is that what we're looking at here are antibodies binding to the hemagglutinin of influenza. And type A virus is the one we're most worried about. This is the one that can cause the most disease in humans. And as far as how close these hemagglutinins are related, there are two types. There's a type 1 and a type 2. So here's the numbers for the different types. Here's influenza B, which is very different than influenza A. And so the idea is make a universal vaccine or a universal set of antibodies that can block or interact with all of these different types of hemagglutinin. So broad protection. So this comes into play when you look at figure 1. Right here, you can see all the different influenza viruses that they are looking at. So you've got B, and then you have type 1 shown in blue, and A group type 2 shown in green. In figure 1, these are going to be your single domain antibodies, and they first then take a look at kind of I call it a mini multi-domain antibody, because they're linking two antibodies that work good against type B together, single domain, and two single domain antibodies that work good against type 1 and type 2. And so they're comparing how well these antibodies neutralize influenza virus. So let's talk about a neutralizing antibody. Oh, sorry. Before I talk about neutralizing antibodies, since this is looking at the same thing, figure 3 is going to show you your multi-domain. So we have the influenza A single domain antibodies, linked together to the influenza B ones. Here's where we put on the FC region of IgG, and here is the control antibodies from an influenza vaccine. This is missing the label of IC50. It's the same scale or units as figure 1. And they're doing the same thing, looking at type 1, type 2, and type B influenza, and how well, now as they're creating these bigger and bigger antibodies, multi-domain ones, how well do they neutralize the virus? Okay, so now we're going to talk about neutralizing antibodies. The key to a neutralizing antibody is right here, that it blocks infection. And blocking infection can mean totally binding to the virus and blocking it from interacting with the cell at all. blocking attachment, blocking entry, so endocytosis in this case, or blocking uncoating. But the overall key is you're blocking infection, so there's no more virus made. Okay. So the question is, how would you know if these are neutralizing antibodies? This is your goal. You can't really see it happening, so you have to measure it. So the neutralization assay is where you mix your antibodies with your virus. And in this case, they let it incubate for an hour and a half. So they're letting antibodies bind if they can, doing it at different dilutions, right, to see how much antibody you need. And then they take this serum after incubation and they add it onto cells that will be infected by any free virus. So cells will be infected by free virus, or you could say non-neutralized. Okay. Allow the infection to go for 3 days. Harvest the liquid, so how much virus was produced came off of the cell, and do a hemagglutinin assay to kind of quantitate how much virus was produced. Okay. So ideally, right, there would be no virus produced, and when you do the hemagglutinin assay, you would have no reaction. So what's happening in the hemagglutinin assay is that normally, so this is your control, you put red blood cells in a little dish, right? So you've got your red blood cells. They're just going to settle to the bottom because they're heavy. And if you looked down from the top of this dish, you would just see this little red dot where all the red blood cells would just sat there. So that's no reaction. If there is virus produced in that liquid, when you mix the virus plus red blood cells, influenza will cause the red blood cells to clump. And so instead of all the red blood cells hanging out, you're now, let's see if I can draw this better, really having red blood cells clumping around and these complexes kind of floating. So you see something different. So you see hemagglutination. Okay. Sometimes, and not in this paper, so I'm going to make this across, but just to explain what's happening. Sometimes they'll look at neutralizing antibodies just in solution to see if they're binding and looking for hemagglutination. This is not what they did in this paper, but it's another part of the HA assay. So when you look back at figure 3 and figure 1, what I want you to notice is that the lower the IC50 number, okay, so the inhibitory concentration for 50%, so how much antibody do you need to inhibit 50% of the virus from being able to infect? The lower the value, the greater the inhibitory activity. Okay, so things that are way up here, whoops, like here, this is telling you that this CR9114 antibody really doesn't inhibit influenza B at all. So this is definitely not a broadly neutralizing antibody. So take that information as you work on analyzing your data. Figure 2 is beautiful crystallography data. The gray is the HA of influenza. And then you can see in the colored where the antibodies bind. So they wanted to analyze their single domain antibodies and ask where they bind. Because remember, the idea is you want it to bind to the stalk to make it more like broad spectrum, working against multiple different types of influenza. So once you've created these antibodies that you know work, okay, they're going to do antibody delivery. So this paper is not about a vaccine, but about immunotherapy, which we talked about in class, is where you give a person antibodies directly. So this is not a long-lasting, but it is more immediate protection, because you're not asking the body to recognize the foreign antigen and to create antibodies and B cells and memory cells. You're just saying, okay, here's an antigen that works. So they did this a couple ways. One is a direct injection of the antibodies. So day 0, they gave the mouse the antibody with IVs, and day 2, they did an intranasal challenge with influenza. Okay. And they wanted to see, do these antibodies protect from a virus infection? And so when you look at figure 4, A through D is this assay. Okay. And I highly suggest you look at figures, supplemental figures 9, 10, and 11. They show about body weight, so they're monitoring the health of the mice, and they talk about viral load in the lungs. So how much of this virus that was in the nasal passage got into the lungs and replicated. The lungs is where we have the most problems, right? You inflame the lungs, you can get complications such as pneumonia. Okay, the other part of figure 4 is where they use a different way to deliver the antibodies. Okay, and this is with something called AAV. So here, antibody delivery is using an AAV vector. So the AAV is a small, non-pathogenic virus. And it can deliver DNA to cells. So now we're not packaging, we're not putting in antibodies directly, we're delivering the DNA that will produce the antibodies via the cell. So the cell will actually do the transcription translation. So that's going to obviously take a little longer. So at day 0, they give intranasal AAV, and then at day 7, they do an intranasal challenge of virus. And the question is, how well does this method of delivering antibodies protect against a virus infection? So for E through G is looking at, how well the mice, or if the mice live. So these are survival curves. So across the top of all of these is 100% survival, and every time you go down, that means a mouse was sacrificed or died, most likely sacrificed due to loss of body weight, bad health. So they're looking at 2 different methods to deliver the antibodies, and challenging with B and type 1 and type 2 A viruses. Just so you understand a little bit about AAV, it has been used quite a lot in gene therapy methods because it's such a nice little virus. You can easily manipulate the genome, and normally you can create AAV to cause integration. I don't believe that's what they're doing here. I need to relook at the paper. If they actually integrated the antibody information into a cell, that would be pretty amazing. The other question I'm not sure about is if all these different, what cell types they might be targeting that can produce these antibodies just as a normal protein and release them through exocytosis. So I'll keep looking at that this weekend. But it's just a gene delivery method. So we're delivering DNA, asking the cells to produce the DNA. So what, at least at this point, I've read through phage display and the methods they used for selecting the antibodies. I understand that they've gone through multiple rounds, but I likely will not be able to explain much more than that. Crystallography methods, if you look in the supplemental material, there are pages and pages describing the crystallography methods. I pretty much look at the pretty pictures and try to understand what they're getting out of it as far as antibody-antigen binding. They talk about ADCC, which is antibody-dependent cellular cytotoxicity, I believe. And it's basically where antibodies can promote more cell destruction, and they don't show this in the main paper. It's all in supplemental. So I will look at this some more to see really what the importance of this is and see if I can explain how they did it a little better. But just letting you know where some of my weaknesses are on this paper. Okay, I hope that helps. I apologize for running through it so fast in class, and I look forward to our discussion of the data and results on Monday. Thanks.